So, Bioengineered Jam Sessions. Self is the project that I will try to talk about and focus at the end. And I thought that it's going to be kind of a workshop-y vibe here. So there's a lot of images and kind of details about how. And I hope that that's not going to be too boring. So as I said, I'm a researcher and an artist in SymbioticA. I'm in UWA. The world, the cultural sphere that I work with is kind of the new media world. I mean, the technologies that I'm using are, I consider them to be emerging technologies. I'm mainly focused on biological technologies and kind of the disciplines that interest me more are neuroscience and stem cell technology, and I'm using both of them quite extensively in my work. And I kind of live in this kind of scale of grow and make. The robotic bodies are kind of more engaged. Me and my collaborators, we make those robotic bodies. But my personal focus in all those projects is growing. I grow those neural networks, or I grow those brains, quote unquote. Most of the projects that I engage with are trying to problematize those biological technologies that we're using rather than celebrating them, problematizing them by putting forward absurd scenarios and contestable futures. I made a mistake here. Absurd and contestable scenarios. And when I get to the work itself, you'll understand more what I'm talking about. So when I kind of, in the last couple of months, when I was thinking about kind of writing a paper, and for a couple of conferences that I've been, the title of those talks are usually the Bioengineered Brains and Robotic Bodies, from Embodiment to Self-Portraiture. Someone in one of the conferences that I was in called me the matchmaker, because basically there's this process of matchmaking between the biological brain and the robotic body. And when I kind of look at the work that I've done in the last couple of 10 years or even more, 15 years, the robotic bodies, the nature of the robotic bodies from aesthetics to function usually are informed by the nature of this brain, the biological brain that we make. So I kind of like the matchmaker metaphor or title. Misbehaving machines, I don't know, I wrote it, but I have nothing really to say about that, because my machines, they do behave. They die at the end, but they behave here and there. But really, when you look at the kind of biological meta, you can't, or biorobotics, you can't really talk about it without just kind of mention kind of the ethics of the technologies that we're using and how problematic it is to kind of make brains, even though, and I'm going to use the word brain as far as terminology, but I have to explain that those are not brains, they are neural networks. In those neural networks, there are 50 to 100,000 neurons that are forming the biological network, compared to a brain that is, and they're grown in 2 dimensions, compared to a brain that is 3-dimensional, highly organized organ that has billions and billions of cells and trillions of synapses. It's quite a symbolic brain. But for the sake of this talk, I'm going to use the term brain. And despite the fact that it is symbolic, the use of those neural networks allows us to kind of bring it into kind of the cultural sphere and get people to think and initiate some sort of dialogue about the future of those technologies, about the possibilities of making brains, about the possibilities of manufacturing consciousness or intelligence, and to generate some sort of an ethical debate about whether we should even go down this path, or if we do go down this path, which, it's far away, but, it's happening. What sort of responsibilities or what sort of ideas should we kind of culturally form around the making? So, some of the projects that I'll talk about, we're looking at embodiment. We gave those neural networks, we gave those brains bodies, robotic bodies. And the last project that I'm going to talk about is a self-portrait. And this is why I was talking about the narcissist experience of mine. So when you talk about biological self-portraits, you can't not mention Michel Duchamp's piece, The Faulty Landscape, where he ejaculated semen on a piece of silk, and that was kind of his self-portrait. And you know, a lot of artists, or there's a large number of artists that try to kind of create or to produce biological self-portraits. I have worked with semen or kind of sperm cells before, but I found it really hard to run to the toilets 5 minutes before the opening and just kind of produce the biological material. It's not recommended. So Justin Cooper used MRI to produce her kind of self-portrait, Marta de Menezes, DNA sculptures, Charlotte Jarvis, which is kind of took some different cells of her body and tried to IPS them and to show them. And that was, I don't know how good you see it, this is kind of a stage in my self-portrait. Those are neurospheres, so neural stem cells of mine that are starting to differentiate into kind of a neural network. But this is not it. It's just a nice picture that I like. When I work with those biological materials, when I'm kind of thinking about the process of making the brains, you know, it really reminds me of the process of, and I'm oversimplifying it, of course, of robotics, where, you know, there's kind of the hardware design, the software design, and a consideration for the sensors. It's kind of the kind of main three issues with robotics. And when I can make those brains, I engage with a similar kind of process. The hardware are the cells. Those are the neurons that grow, the neurons that produce, that make the brain, that make the neural network, that produce the data. There's the software. I'm using stem cell technology called IPS, induced pluripotent stem cells, where you can take an adult cell, skin, or whatever it is, and you can reprogram the cell to go back in time to be a stem cell, and there you can differentiate the cell to whatever other cell you want in the body. So basically you're playing with the genome, you're playing with the software of the cells to create the function that you want. And then the sensor, the interface, I work with a multi-electrode array. This is kind of a specialized Petri dish that has, also pictures later on, or that has 64 electrodes that are fitted at the base of the Petri dish, and the neural network grows on top of those electrodes. Those electrodes are a read and write interface to and from the neurons. The electrodes can listen or record or get the data that the neurons are producing in 64 areas of the dish. And at the same time, those electrodes can stimulate the neurons, send information to the neurons in 64 areas of the dish. That's a high magnification image of this array. Those are the electrodes. The neural network, as you can see, grows on top of that. These days, there are higher density arrays, but we're working with this 64 one. That's how the dish looks. I'm collaborating with a scientist called Steve Potter. He has a lab in Georgia Tech in Atlanta. And I've been in his lab a couple of times, and our collaboration is quite close. He's looking at those neural networks, and he's trying to kind of see whether whether he can detect any signs of emergent behavior or learning in those neural networks. And when we started to work together, I kind of piggybacked on those experiments and tried to embody those neural networks with our robots. Just kind of a quick video shows kind of the neural activity on the dish and how it responds to simulations. So it's a plot of 8x8 electrodes, spontaneous activity, and the way the neurons react to stimulations that were sent to the network. This is another way of looking at it. See again, a plot of 64, it's really 60, 64 minus the corners. And the long kind of things are the action potentials. Okay, so the first project of Embodiment, and I'm just going to go really fast on those projects. If you want to ask more or get more information, I'll tell you later on. First time that we embodied one of those neural networks with a robot was MIARC. We gave it, scientists usually kind of use utilitarian kind of tasks, when they kind of look at data. We decided to give this robot the least utilitarian task that one could think of, and that's being an artist. We asked those neural networks to respond to stimulations the shape of portraits. And really, one of the ideas that really interested me was to try and engage and communicate with those neurons through the feedback loop, through the two-way communication of the dish. So that was a drawing arm, 3 pens on paper. The camera was kind of looking at the progression of the drawing and sending to the neurons. What an interesting thing about this work is that the neurons were in Podo's lab in Atlanta. The arm was wherever we were in the world, and the communication was done in real time through the internet. So it's some sort of a geographically detached, semi-living entity where the kind of the body and the brain work in different continents and time zones. After that project, we produced Silent Barrage. Again, same architecture of the dish in Podo's lab, but that was a different project where we kind of created an immersive environment. I don't know how immersive it was. Kind of an environment where we invited the viewers to walk inside this robotic interface. We had cameras that kind of detected where the people are in the space, and this information was sent to the neurons, basically getting the people to communicate almost, not in a direct way, but to communicate with the neurons. And it was really interesting to kind of see how people behaved in this space when they understood that they're actually talking to neurons. Another kind of layer was the scientific experiment that was done there. Those neurons that we were working with experienced epilepsy kind of behavior, epileptic behavior, because this is how, and we used those stimulations that were generated when people were in space to try and calm those epileptic seizures down. And poetically it worked really well because we invited people to walk into this space and to try to cure the networks from their epileptic fits to silence the barrages, and hence the name Silent Barrage. And then I learned about the IPS, Induced Pluripotent Stem Cells technology that allows us to reprogram edited cells and produce any other type of cell in the body. Well, take them back into stem cells and then, and I was really interested in kind of just playing, and this is kind of the way we work in SymbioticA, and I'm sure in a lot of other places. It's all about speculative research. It's all about being playful with the technology. As I said, trying to problematize the technology by putting forward absurd scenarios. I had two stories about misbehavior in the last two pieces, but we leave them to the end, maybe. Just gonna remember it. Anyway, be playful with the technology, try to kind of muck around with that and speculate about it, problematize it. So we came with this project that got the nickname Project Dickhead, where we basically purchased foreskin cells in an online catalog. We grew those foreskin cells, and then we IPS them to make a brain. The official name was kind of in potentia because the funding body said that if I won't change the name, I won't get the money for the fellowship that was sponsoring this project. So we changed the name from Project Dickhead to in potentia, and this is how it kind of looked. The neural network was here, and in this project there was no interaction. We didn't ask the brain to do anything, just to be on the pedestal. We put the brain on the pedestal in kind of in a way to critique the idea of the importance of the brain in the Western society in the last 50 or 100 years. And kind of the questions about what is really alive. Today, the question of what is alive is measured by the how much our brain works. And we do kind of refer to this project as a robotic project in a way, because we did put the neural network on a pedestal, a robotic pedestal that actually kept it alive for a month's time. So in case this pedestal had inside an automated biological lab with an incubator, with a fridge, with that, it kept the atmosphere, and fed the cells and cleared the media. So it was all automated. So we had this, I engaged with those projects of embodiment where we kind of grew the brain that were made out of kind of mouse cells and gave them robotic bodies like Simon Barrage and Niart. And then there was here the kind of being a little bit playful with the software of the cell, creating a brain, making a brain from human foreskin cells. And then I decided, okay, so this is when I decided to kind of go for my self-portrait. And I went to the hospital, as you can see here. And I had a little surgery or biopsy, it's not a surgery. Just got a little piece of skin out of my arm. So, it was quite an interesting experience to kind of run with the, with the tube and the kind of biopsy to university, just hoping that I'll get kind of parking because I had 1 hour for the cells to kind of be viable. So I did the Israeli thing and I double parked and ran to the lab. So this is the piece of biopsy, grew them in the lab, just kind of, those are my skin cells growing in culture. It's just, the movie was done over 12 hours. You can see how the cells divide. Over here, they kind of lift up and divide. I'm just gonna, despite the fact that it's a crowd pleaser, I'm going to continue because I don't have a lot of time. And then I kind of took those cells and I shipped them to the University of Barcelona to work with a colleague of mine to IPS and reprogram those cells. It was great to work in a lab next to the lab that Santiago Cajal, the first scientist that actually observed those neurons in vitro and made those amazing drawings in the 18th century. He was a great neuroscientist, and the lab was just next to the lab that we worked with. We reprogrammed the cell, and by the way, when I took half of the quantity of the cells that I had, froze them, and shipped them to Barcelona. And I woke up the next day to get an email from the shipping company to say that they just, the cells were kind of, because of negligence of the courier, all the cells were dead. So kind of felt that half of my mass kind of died on the way. DHL, be careful, don't use them. Anyway, so in Barcelona we produced those stem cells. You can see that the stem cells are the cells that are kind of growing in clumps in the middle. The cells around them, it looks like a brush strokes. They're kind of the feeder layer cells. They help to the stem cells to grow. And we produced quite a lot of colonies of mine. It's me in the lab. That's the money shot of bioartists. And then we took half of those cells and we froze them, and which is kind of an interesting thing as well about kind of freezing life and how do we freeze and how do we thaw life. And we sent them to UWA, where we pushed the stem cells into down the neural lineage and created neural stem cells. And now, until 6 weeks ago, I've been struggling with kind of differentiating those neural stem cells into neural networks on the multi-electrode array. Those are just some images of the differentiated neural networks. Those are the kind of massive kind of neural stem cells. They kind of, they're called neurospheres, and then when they differentiate, they start sending processes around. You can clearly see the kind of neurons growing out of this kind of mass, and a few kind of fluorescent images that kind of, with fluorescent dyes that confirm that they're neurons, more or less. And that was the first culture that actually produced electric signals for me. It took me literally billions of cells that were my cells that were cultured under, hundreds of cultures, 9, probably 12 months with 11 different protocols. I mean, the protocols to do what I really want to do are not there. So I work very close with the scientists to develop our own method that we're going to publish very soon in the scientific magazine. And after 12 months, we managed to get signals from those two electrodes. And now we're kind of optimizing we're optimizing the techniques of producing and making those brains and getting them to be viable from an electrophysiological point of view on those arrays, which is quite a hard thing. So this is kind of this brain of this self-portrait of mine, and then I started to think, so what sort of body would I give myself? And that's kind of a nice question to ask. If you could, what sort of body would you give yourself? And I'm not really into the humanoids, I have to admit. I prefer the robots to be kind of a little bit more abstract and get away from a humanist kind of depiction of bodies. But kind of thinking about how do I portray myself? I didn't really want to kind of go hard on this question. So I decided to kind of look at my past, at kind of the card of dreams, kind of my juvenile dreams when I was 14 years old. And that this is what it was. Sex and drugs, it's not a problem. I don't need robots for that. But rock and roll, that's a problem because I can't play, can't make any sound. So I decided to give a sound producing body, to make a musician to give a sound producing body to the brain that we made. And we decided to kind of embody those neural networks with modular analog synthesizers. And it kind of fitted in so many ways. It fitted in so many ways. One of them was the aesthetics. The aesthetics of those modular synthesizers really reflect the aesthetics of the electrophysiological labs in the university. So, there's a correlation there, but also, in a really strange way, the ways those synthesizers work with the control voltage that go from one to the other kind of components reminds me of the way kind of the neural network works, with the way kind of the data is passed between the components. So, and then kind of when we looked at the history of electronic music and kind of a few kind of references to kind of start from there, we saw the kind of neural network, the artificial neural network synthesizer that David Tudor, the guy that played with John Cage, made in the 90s. And in one of his papers, he's talking about the fact that his synthesizer is controlled by an artificial neural network, but the next step up is to make the organic one, the biological one. And that's exactly what we're trying to do. So we're talking about the feed neural network. The signal would go through bracket box to signal amplifier, synthesizer, and feedback stimulation. We're keeping the whole system analog. There's not going to be any computer on the way. As far as musical influences, we don't really have time for that. This is one of the early suggestions. By the way, Nathan, that is working with me on the design of the object. Well, he's working on the design of the object, is here. This is one of our early propositions of how it's going to look. It's not going to look like that. But I think what's interesting to show is that the modular synthesizers are going to be embedded in one object with the incubator, so it's going to be all in one. And the idea there is to have jam sessions. So we're going to invite or curate musicians to come and play with Self, where the human music is then going to be streamed into the neural network through the interface. The neural network is going to respond to the stimulations that were sent through the array, control the array of synthesizers, and to create a feedback loop, or really a jam session between the human and the non-human musicians. This is our first test that we made a couple of weeks ago or months ago. These are not my neurons, those are mouse neurons that we used before we managed to get proper signals from my neurons. And we are using information from one electrode. Without without any consideration for the sound or the object aesthetics.
Yeah, we're wasting money on EEGs now to try and make it's not real.
The video is not running for some reason.
It's not on and off. But this somehow fits into the discussion about it is about activity being part of the listening. Kind of a
all those sounds were triggered by action potentials within the neural network that were recorded on one electrode. And now we're working quite hard on the idea of mapping the data from 54, 59 or 60 electrodes to the neurons. And everything in the system is going to be controlled by the neuron. So, of course, the sound, but also the specialization of the sound. Because when we talk to the neuron scientist, I mean, the neural activity is reflected in two ways: the temporal activity of the neurons, and then the spatial organization of the neurons within the dish. And both the spatialization of the sound within the space, and of course the synthesizers are going to be controlled solely by the neurons. And Nathan promised here to automate. To kind of get the knobs on the synthesizers to move automatically, very similar to kind of old automata and kind of the pianos that play without the player. Yes. Yeah, no question. It's another thing for the list. So it's a big team that is working on this project. Darren Moore is a sound artist from Singapore. He's the head of music in La Salle College. Andrew Fitch, he's a magician from Perth. He's an electric engineer and a synthesizer builder. He's really amazing. And he has a brand of synthesizers that he's developing called nonlinear circuits. Nathan is here. Stuart Hodges is a neuroscientist. Douglas Buckham is a neuroengineer from Switzerland. And Mike Adele is the stem cell person. And that's it.